Hi again, everyone. In this video, I hope to kind of wrap up this series of what I've been discussing and kind of look at synapses, which is the major portion of neurons that we have yet to cover, and then kind of review a lot of the aspects of what we talked about and kind of help with synthesizing the information. So let's jump right into it. Okay, so as you'll remember, this is our friend the neuron, and just to quickly review some aspects of anatomy, we have the dendrites, which are responsible for receiving information, soma, the cell body, we have the axon hillock, we can see the myelin, and we're actually going to focus a little bit more on these end processes, the axon terminals. Now, if you were to the entire purpose of this portion of the neuron is to connect with other parts of the body, whether it be other neurons, directly onto a muscle, wherever it might be. This is the portion that kind of communicates the electrical signal to other places. Now, this area, that connection, is called a synapse, and it's very important to understand that connection, because that kind of determines the integrity of that connection, determines how well the information is going to be conveyed. So in case you're a more visual person, if you wanted to see, basically, if there was another neuron here, this could be another dendrite, and you could see that this would be attaching to another dendrite and synapsing there. You can see this is the reason why I have my pictures drawn beforehand. But hopefully this gives you a little bit of a visual understanding of what's actually going on. So remember, the impulse is going to start here, travel down, and it has to somehow get to the other neuron. And there are two ways it can do that. It can either do that electrically, or it can do it chemically. Now, the electrical is quite simple. They're basically attached through something called gap junctions, and the current just goes straight on through. The more complicated one is the chemical synapse, and that's what we're actually going to talk about today. So I'm going to leave this up for just a second so we can kind of take a look at what we're looking at. So this is the presynaptic neuron, and this is the postsynaptic. And if you're a little bit confused as to what we're looking at right now, this is just a very zoomed in picture of this interaction, that interaction between the green and white. So this postsynaptic part is part of this neuron. It's the dendrite of this neuron. And this presynaptic is still part of this green original neuron that we had. So what we're going to do is talk about how a chemical synapse works. Now, before we jump into it, we have to realize that a chemical synapse relies on a chemical, and that chemical is called a neurotransmitter. And there are many different types of neurotransmitters. You may have heard of some of them, for example, acetylcholine, which I'm going to abbreviate that way, norepinephrine, GABA. It really depends on where the neuron is and what its function is, whether it's stimulatory or inhibitory, to determine what neurotransmitter will be there. Now, its neurotransmitter, just to reemphasize, is basically just a chemical that is stored in the presynaptic side, so it would be somewhere down here. And they're stored in membrane-bound vesicles. And the requirements to be considered a neurotransmitter is all that, and the only things that it has to have is that it has to be present in the presynaptic cell, it has to be secreted upon excitation, and it has to activate specific receptors on the postsynaptic neuron. Now, let's delve into how this process works a little bit more. So just to reemphasize, this is the presynaptic side, the postsynaptic side, and the depolarizing current is going to come from up here. So that's as if it was traveling all the way down, came down, this side over here, and it's eventually, when you're zooming in, ends up over here. Now what's going to happen is this depolarizing current is going to eventually activate this ion channel. And what this is a voltage-gated calcium channel. And when it is opened, it allows calcium to flow in. The inward flow of calcium causes some changes to happen in this presynaptic neuron. And the main thing it'll do is it'll cause these vesicles to bind to the presynaptic membrane. So I'm going to show kind of like a time lapse. So let's imagine the same one. It's going to move a little bit closer, and then it's going to actually be exocytosed, which means it kind of merges in and releases its contents out into the synaptic cleft. So let me redraw the fact that there's still neurotransmitters in here, and it's going to merge, and it's going to kind of plop these little contents out into this area. This area is important. It's called the synaptic cleft. And it's actually a physical space that exists between the two neurons. And that's the difference between chemical synapses and electrical. Electrical synapses, there's not really a space that exists. And those neurons are the kind that need to communicate very, very quickly. They're important for, having similar actions occur at the same time. Chemical synapses are important in things like learning and memory and such. And you can also see chemical synapses between neurons and muscles. That was a quick aside. So now we've seen that we secreted, or excreted I should say, these neurotransmitters into the synaptic cleft, and they're going to traverse this very small distance and attach to receptors on the other side. Now there are two types of receptors that could exist on the postsynaptic membrane. There can be metabotropic, and all that basically means is that these receptors are things like G protein coupled receptors. And if you don't know what those are, you can learn a little bit more about them through other resources, but they basically activate like metabolic processes within the cell, and they kind of take a little bit longer to affect changes within the cell. Or the neurotransmitter can also bind to ionotropic receptors, and these cause very quick changes. They can cause inward flows of ions such as sodium. And if you'll remember, sodium is a positively charged ion, and that can affect the membrane potential of this postsynaptic neuron. And just to make a connection with our previous lecture, let's assume we're looking at a graph of this particular postsynaptic neuron. Now, before anything binds to it, it's going to be at -70 millivolts because it's resting, right? Now, let's say the presynaptic membrane releases a neurotransmitter. It binds to this, it opens up this channel, and allows sodium to flow in. Well, that counts as a stimulus, so it's going to start depolarizing. Now, if sufficient sodium enters, guess what's going to happen? It's going to have an action potential. And once there's an action potential, it'll travel down this neuron, it'll end up in the axon terminal of that neuron, and it'll do the same thing with the neurons that it connects to. So you can see a nice little chain of events that connects all of these together. Now, it's important to note that there's not always just a depolarizing effect. Sometimes you can have channels, I'm just going to use an arbitrary color, you can have some channels that when the neurotransmitter binds to it, okay, it can allow negatively charged ions to come in, not positively. And if negatively charged ions come in, guess what happens? It doesn't depolarize the cell, it hyperpolarizes it and makes it harder to fire. Now, this concept is important to remember because you don't just want to always fire neurons, you want to be able to modulate it. And the way you modulate it is by having a way to fire them and a way to stop them from firing. So this is an important concept. Now, the last step that occurs that we have to remember is that you can't just have this these neurotransmitters exist in the synaptic cleft forever. They have to be reuptaken somehow. And one of the most common ways is there's an enzyme that will help reuptake the or to degrade this substance and kind of take it back into the presynaptic neuron. So just as a quick review, and we can write it out real quick, there are a few basic steps that we should remember. First, there has to be biosynthesis of neurotransmitter, which is going to happen in the presynaptic side. Secondly, there's going to be storage of neurotransmitter, which we see in the vesicles here. Third, there's going to be release of neurotransmitter, which we know happens when there's a depolarizing current and it gets released. And we know it's based on the voltage-gated calcium channels. Fourth, there's going to be transmission of signal, which we know happens whenever the neurotransmitter releases and binds to receptors on the postsynaptic side. And finally, there has to be reuptake or breakdown so that the signal doesn't last forever. and that happens through enzymes, and sometimes there are even cells such as glia that can help take that up, but those are just specifics. So I hope this video has helped you understand how a lot of these things function together. We've been able to talk about everything from the very beginning, starting from an action potential being created, propagating all the way down the neuron, and ending up in this region where it can continue the action potential to other neurons and other connections that it has. I hope this has helped you understand a little bit more about the basics of neuroscience. Thanks.